So hi, this is Clayton Thomas. I'm the founder of Root, and I'm really, really excited today to be able to share some time with really the leader in the world of research around glyphosate and uh, agriculture. So Dr. Don Huber, I'll give you a brief overview of his history. He's a professor emeritus of plant pathology from Purdue University, and I've followed his work for about a decade, being probably the leading researcher on glyphosate. He's a PhD from Michigan State. He's a graduate of the U.S. Army Command and General Staff College and Industrial College of the Armed Forces. You've published over 300 journal articles, spoken at over 84 special or done work with over 84 special invited publications. You've testified at The Hague regarding glyphosate and its issues pertaining to Monsanto. And you've spent about 50 years in your space as a proponent of glyphosate and how to clean up soil or best ways to work with agriculture and development, correct?
Well, I've had 60 years of research on soil-borne diseases and microbial ecology in that area, but been working with glyphosate since 1974. So since it was registered as an herbicide, I started seeing things that we couldn't explain from the information that was being provided by Monsanto and other areas. And so just from a curiosity standpoint, I got started because we were seeing the increased severity of a number of diseases that I thought I had a pretty good understanding of that didn't fit the picture relative to the information that the company was providing from an herbicidal standpoint. And so one thing led to another, and the deeper you go on this chemical, the more concerned you get.
So can we dive into that a little more? Because you are the guy that's been at the forefront of understanding, the implications of using this as an herbicide. And with your experience, you saw the correlation between the transfer from plants into humans really early on, correct?
Correct. And that's because it's a very broad spectrum mineral chelator. Of course, minerals are both components of plants and animals, and that's what our body's built from. And so when you have a broad spectrum mineral chelator, those chelators those minerals are also the cofactors for your enzyme systems. They're the catalysts that are the keys that turn that whole powerful enzyme function on, so that when you look at all of our herbicides, they're all mineral chelators to pull that key out of the ignition or to immobilize that mineral so that it's not physiologically active. And you see that same function in animals that you see with humans. So I started seeing that broad spectrum mineral aspect. We started seeing the reduced nutrient density of our crops that we were producing. But then glyphosate has another aspect to it that compounds that dramatically, and that's the fact that it's actually a very broad spectrum patented antibiotic. It's an antibiotic, though, that is toxic to the beneficial organisms in the soil, in our crops, and in the GI tract, but it stimulates the pathogens. And that's what first got my attention, was the increased severity of several of our plant diseases that were associated with the mineral ions, as well as with the biological controls, natural biological controls in the environment that were being decimated by this herbicide. That same function or same antibiotic activity then is what induces the gut dysbiosis, the inflammatory bowel diseases, the all of your gut-brain functions. That tie is all based on a balanced microbiome. So what you see is a disruption of the entire biological system that we rely on as a society or as individuals. So one thing led to another, and again, as I mentioned, the deeper you go on this chemical, the more concerned you have to be.
And this, so if I understand what you're saying, I mean, when people are familiar with antibiotics, they think of killing the good guys, or the bad guys, you know, from a system perspective. But what you're saying is this is inverse. It actually destroys all the good guys, and its intention, whether it could be direct or not, the intention was to kill or to prevent crops from being damaged by bacteria or some of the issues on the plants. But would you call it an unintended consequence, or it's just a direct consequence of this destroying everything good in us?
It's a direct consequence. Actually, that's the herbicidal mode of action of glyphosate. You can't kill a plant with glyphosate in sterile soil. What glyphosate does is give that plant a bad case of AIDS, essentially as an analogy, so that it becomes extremely susceptible then to all of the other organisms in the environment that could be pathogenic. Excuse me. So we see that Fusarium, Rhizoctonia, all of those soil-borne pathogens are the organisms that really kill the weeds. What the glyphosate does is remove the resistance mechanisms through that shikimate pathway and also several other pathways. Glyphosate actually downregulates 291 enzymes. So it's not just the EPSPS enzyme that's affected, but all of those enzymes are involved in resistance, so that when it shuts those mechanisms down, then it leaves the plant very susceptible to the pathogens. Excuse me. And it's the pathogens that do the killing. That's why it takes 4 or 5 days or a week to kill the plant with glyphosate, where a contact would do it overnight. And so it leaves it very susceptible, but it stimulates the pathogens. So even some of the organisms that are very weakly pathogenic are, many of them not even of practical significance economically, become very serious pathogens for us. An example from plants would be Cornospora. It's a root rot on soybeans, but before glyphosate, we never gave it any credibility as an economic problem. We saw a little root nibbling once in a while. The plant would respond, and it was kind of like getting a runny nose overnight, and then the next day it was gone and you didn't worry about it. The other thing with glyphosate as an antibiotic, as Jack Heinemann in New Zealand has shown in his research, is that you not only develop resistance to glyphosate in an organism, in a pathogen, but it also induces resistance to all of our other antibiotics. So that our concern about the loss of antibiotics for human and animal use for controlling pathogens is in large part also due to glyphosate, because it induces susceptibility or increases the virulence of Pseudomonas or Streptococcus or other organisms that normally wouldn't be stimulated, to make them resistant then to penicillin or tetracycline or cephalosporins to our other antibiotics. Again, the deeper you go in studying this very simple chemical, the more concerned you get, because it's literally does what it was designed to do, and that's to kill.
And it doesn't in human after we've consumed the plants that have been sprayed, and it's destroying us just as well as it destroys plants. It's just taking a little more time.
Right. It's a very persistent chemical in the environment. U.S. Geological Survey and looking at how much glyphosate's in soils. Some areas in the Midwest and Florida will have over 2 tons of glyphosate per square mile residual. It's been building up for 30 years now from repeated application. Very difficult compound in most environments to degrade. Yet clay content is one factor that will give it persistence, so that in a good clay soil, lower pH soil, it has a half-life of over 20 years. So I was looking at some of CSIRO's data from Australia here last February, a year ago, and they can account for 100% of the glyphosate still residue in the soil that's been applied for the last 20 years. Essentially zero breakdown beyond the AMPA level. So we have a residual or a persistent aspect to it. And then it's very broad spectrum interference with all life processes, essentially.
So when we look at soil, because you've been doing this, I think since you said 1974, how much of the farmland in our country, whether it's organic now or not, because that transition has happened, say over the last, we'll give it 20 to 25 years, we'll say 20 years, that farms have tried to go organic. How much of the farmland in this country do you think has had exposure to glyphosate, or still has glyphosate on it, even if it's not being used currently?
Probably 95%, other than pasture ground. You can find some pasture ground, you can put out experiments. The university, or Guelph University, tried to repeat Miriam Fernandez's research, showing that one application of glyphosate 3 years previous to a wheat or cereal crop would predispose that crop to fusarium head blight and the toxins that fusarium produces, the T1, T2 toxins, azalerone. They couldn't find any soil in their province that didn't have residual glyphosate in it. To repeat her study that she was able to do in Saskatchewan, where she could start out with non-glyphosate soil and then set those experiments up, so that we have a tremendous contamination factor that's involved from a research standpoint. Excuse me, I got a dry throat here.
As long as it's no viral issues that are causing issues with your respiratory system, we're good.
I made the mistake of eating some things that increase the mucus, and shouldn't have done that.
So you made a comment a minute ago about the antibiotic resistance. So based on your research and your knowledge, with the rates that we've seen for methicillin resistant Staphylococcus aureus, do you think that there's a drawn correlation between our issues with MRSA and our exposure to glyphosate now? Because it's a massive issue.
I think Jack Heinemann's research says there's no question. But with the heavy exposure to glyphosate, we're bound to have resistance to most of the other antibiotics.
And I definitely want to get on into some human questions, but when we look at our feed animals, whether it's, you know, chicken, beef, pork, they're grass fed, hopefully, but the majority of their food supply is sprayed with glyphosate. So the chelating aspects, if you have chicken that are eating corn that's been sprayed, or grass or other components for other feed stock, the chelation aspect in a feed animal and their exposure, and then our consumption of those animals, has there been much research to look at the transference in the meat supply as well as in the egg standpoint?
Yes, it essentially bioaccumulates in all tissues. So that, but from an animal production standpoint, you see the tremendous losses that we take in our animal production because of the antibiotic activity, as well as the reduced mineral content and nutrient density of the feed. In ruminants, we see chronic botulism as a very serious problem. In the US and Canada, we've had bankruptcies in our dairies as a result of high death loss from chronic botulism. Well, our veterinarians haven't recognized that in the past. We're familiar with the acute botulism. That's where you get a possum or a rabbit rolled up in a 1,000-pound bale of hay or something, and then under those anaerobic conditions, you'll see the Clostridium botulinum producing those very serious bot neurotoxins. What happens with glyphosate, as Dr. Monica Kruger has shown in her research at Leipzig University, is that less than 1/10 of a part per million glyphosate in the feed, you end up with gut dysbiosis. You kill off all of those organisms in your gut microbiome that would normally control your Clostridium and your E. coli and Listeria and Salmonella, because its antibiotic activity is there. Then you get gut dysbiosis with an overrun of your Clostridia and Salmonella and these pathogens. Well, Clostridium produces the bot neurotoxins, and she's documented that in her studies, so that it's just a matter of time before the animal dies. Our normal lactation, or average lactation for our larger dairies, is less than 3 lactation periods. Well, a cow doesn't come into maximum milk production until 4 or 5. The reason why we only have a little over 2 to 3 lactations is because their health starts deteriorating so rapidly from the chronic disease problems that they can still make it by selling them for hamburger rather than continuing to milk them. And if they get a downer cow, a cow that finally goes down from the infection, then they lose it entirely. They're not able to salvage. So, so rather than take a chance, they cull out the herd usually after that 2nd or 3rd lactation, and tremendous losses. Our veterinarians are starting to recognize some of these diseases, the hemorrhagic syndrome, duodenal hemorrhagic syndrome, and that system that's very common in poultry as well as in dairy and beef. So those losses in the past would just be unexplainable, and a grower would go out of business, or he'd go out of animal production and become strictly a crop producing enterprise rather than a diversified farm operation. But they're becoming better. Dr. Ted Doopmeyer has a regular update on his regular update on his encounters as kind of a regional veterinarian in Canada. And it's rare that he doesn't have 4 or 5 cases that are nearing bankruptcy as a result of glyphosate toxicity.
So
I may have to take a break and try and drown a frog here, but
go ahead and grab some water really quick.
If you let me do that, a little better.
Hey, there we go. Okay, so you've seen the correlations between how glyphosate is destroying our food supply on the animal side, then, and is there much else based on the research you've done individually and what you're seeing around colleagues like you're talking about from New Zealand and other areas? Is this one pesticide really at the root of all of these issues, the problems we've seen in our food supply, the degradation of our of meat, of milk?
Well, it's so extensively used. When you have 500 million pounds a year dumped into the environment, about half of that to agriculture and half of it in parks and recreation and forestry, that there isn't anything that's missed. So that it's by far the most extensively used agricultural chemical we've ever had by multiples of intents. So that its persistence, its broad spectrum activity, just cover every area. Now there are other products out there that would be similar. Glyphosate or Liberty is its herbicide name, as a sister to glyphosate, another phosphite type of a material. You have your organic phosphates also that will have similar activity against mammalian systems or animal systems, not as broad spectrum antibiotic activity. Don't know of any of them that have the antibiotic activity as extensive as glyphosate, but they're all mineral chelators, and the organic phosphate and phosphites are broad spectrum chelators. Most of our herbicides like Tordon or the Phenoxy chelators, your Puma Gold, all of those herbicides are fairly specific, but they would be specific for just primarily copper or iron or zinc or manganese. Glyphosate hits all the cations. So all of those essential mineral elements that we rely on and our animals rely on for physiological function, as well as for the plant, are all compromised. Our nutrient density is reduced, and our pathogen virulence is greatly magnified in this process.
So you basically come into the issues that we see in human health now, that our mineral deficiencies and a lot of our health issues are directly tied to our exposure, or even our mother's exposure, because a lot of that being passed through the placenta. But our mineral deficiencies aren't due to deficiencies per se, it's due to just being toxic from glyphosate and other metals.
Well, it's both, because we've gone so heavily on the GMO crops. One of the things that happens with that old frog didn't drown, but one of the side effects of disrupting that genetic code is changing the spatial relationship between the genetic materials, and one of the consequences of that is reduced mineral uptake efficiency in the crops, or mineral absorption in the animals, so that we have both a reduced uptake and content, as much as a 50% reduction in many of the essential minerals in our GMO crops compared to the isogenic parent. And then we're applying this a very broad spectrum chelator that is a second whammy as far as the availability of those nutrients.
So what percentage of crops, and I want to ask you, when we talk about exposure to crops, which crops are hit, but from a GMO standpoint, if we look at what consumers are getting in their diet, how much of those are GMO, and what sources are they? Are we talking all vegetables? We can say soybeans, corn, wheat. We know those, but how broad is this issue?
Well, the issue becomes even broader, not just from the GMO herbicide resistance or tolerance level, but also because glyphosate's used extensively as a ripening agent, as a desiccant for many other crops. So that one of the crops that we promote as being very healthy nutrient-wise is chickpea or garbanzo beans, yet almost probably 99, if it's not organic, it's probably been desiccated with glyphosate and has one of the highest levels of residual glyphosate in that bean or chickpea of any of the crops. So even more than a lot of our GMOs that we spray, because we spray the GMOs during the regular growth period rather than at that late stage to kind of force it to die. But the only place that the glyphosate can go then is right into the seed. Well, that's what we consume. And so we get hammered in a number of different areas, different ways with glyphosate. Why it was ever approved for use as a desiccant is beyond my imagination. You'd almost say there had to be a lot of dollars exchanged to permit that, because it's a highly persistent material, water soluble, and systemic in the plant, so that it goes anywhere or throughout the plant. About 20% moves throughout the root system. So you're moving into the soil. Most of those roots are going to be well below an area where you'd have any opportunity for degradation also. So that adds to the persistence of this very toxic, chronically toxic compound.
So if we look at that from a human perspective, then, so we're getting this through our animal products. We're getting it through the milk supply. We know that soybeans are sprayed heavily. So we know that oats are also sprayed heavily after the fact as a desiccant, correct?
Right.
And
right.
So if this chemical is persistent and able to circulate through every part of a plant, the assumption could be made, because we're not too far off from plants genetically. And I think the assumption for many people is when we look at glyphosate, it stays in the gut, and that's not true, correct? It's going everywhere.
Oh, it's a highly mobile entity in the animal, so it accumulates in the brain and the bone marrow, all of our organs, the heart, liver, kidneys, pancreas, all of your tissues, your muscle tissue. It's anywhere the blood goes, glyphosate will go. And it bioaccumulates then in those tissues. So that if you're doing a hair analysis, you can get about a month's accumulation in that last inch of hair growth before the haircut. Give you a pretty good idea of what your dietary intake is. But it accumulates then in the other tissues and gradually builds up. So if you look at the 6 criteria in our FIFRA law, or rodenticide and insecticide pesticide law, of materials that shall not be registered, glyphosate violates 3 of those 6 prohibitions. It's just that when they registered, they said, well, glyphosate only inhibits the EPSPS enzyme, and since mammals are the only living thing that don't have that particular enzyme, they convinced the EPA that it can never be toxic to mammals, to man and animals, so we don't need to do any toxicity study. Well, as I mentioned earlier, it downregulates 291 enzymes, it upregulates about 30. Many of those 30 enzymes that it upregulates are triggers for our disease organisms to increase virulence.
So they were able to focus on one, and it's kind of like being the magician with the trick. You know, watch this, don't watch over here. We're okay with this one. Don't mind the other 330. The one you think is bad, you know, it doesn't apply, but don't look at the trick over here.
That's why in the review on glyphosate in 1995, 21 years after it was registered as an herbicide and used, commercialized, that they still stated it inhibits the EPSPS enzyme, but actual mode of action is unknown. Well, they knew that its mode of action was to increase susceptibility to disease. And that's how it was the organisms that were killing the plants, not the glyphosate directly.
So, and that's what's leading to, you know, the issues we've seen over, well, really the last 30, 40 years, as you're mentioning. So when we look at the bioaccumulation aspect that you talked about in animals, we're really seeing the same thing multi-generationally now, to where our youngest generation, if we look at ADD, ADHD, autism, we can look at cancer risk, all of these risk factors that are exponentially increasing from a generational standpoint. The bioaccumulation that is being passed from mother into child and then going forward is compounding, as well as our exposure on a daily basis. Is that your...
Yeah, no question. You look at Nancy Swanson, Dr. Swanson's study with taking the CDC data on incidence of 32 diseases, and then the direct correlation with exposure to glyphosate in corn and soybeans, you'll see a 0.9 to 0.98 Pearson's correlation coefficient. Anything over 0.46 is considered a red flag for causal relationships. So, you know, we're not quite at one where it's an absolute, but we're so close. And when you get 32 diseases that follow that same curve, an identical curve, essentially, it's not coincidence.
Yeah, I've had a great focus, you know, for the last 16 years, looking at the heavy metals with mercury, cadmium, arsenic, lead, aluminum, cesium, strontium, and such. And now looking at the correlations, you know, of glyphosate, you know, over the last 10 years and its impact. Dr. Stephanie Seneff, and you probably have done some work with her, she's really drawing the correlation between aluminum, mercury, and glyphosate being the big thing for all of our neurotoxicology issues. You'd probably agree with that?
Yes, no question. Stephanie and Anthony Samsell have done an excellent job of connecting the dots biochemically between the changes with the GMO and changes with the glyphosate, then and the physiologic changes that we see with many of these diseases. The other correlation that we see, and not just correlation, but the actual direct activity, is that antibiotic effect for 16 of those 32 diseases, that the best cure that we have is a fecal transplant. And that's about 95% effective for things like autism, inflammatory bowel, difficile diarrhea, and a whole whole series again for that 16 of those very critical diseases that are approaching epidemic proportions for us. That when you restore the gut microbiome and that function through a fecal transplant, then you see complete change in disease expression. But the doesn't take very much glyphosate in the diet to disrupt that whole balance that's established in the system there to create many of those diseases. So
I'm going to lead you with a question on that, and it's going to get into where I want to move, because I'm one that, you know, I love to talk about solutions and find solutions for problems. And, you know, there's such a huge conversation now, and this is a dream to be able to have a mind like yours that we're able to converse over, because you're the guy. So I'm getting really excited. But if we look at fecal transplants and, you know, the benefit, because that can help restore the microbiome in the new host, but with our continued exposure to glyphosate, is that like a negative feedback loop where you can try to put good stuff in, you regenerate, and then you've got all the bad guys coming back in again through diet and through exposure, that it's going to knock it out again, and we just got to keep through that process? And that's going to lead me to my other question.
Well, some people, some of the doctors feel that you need essentially keep on a continuous detox program. The question is, how do you detox? Can't get that frog out of there.
Yep.
Again, with our high dietary exposure, that it's a continuing type of a system. My son and daughter-in-law and I all had a kind of, or another test for urine or hair for glyphosate from a dietary exposure standpoint, and they watch their diet very closely. It's all organic, all very, and a lot of it is grown. They have a good garden, and so they know what their exposure levels are, and it's very low. For mine, where I'm traveling a lot, I don't have that opportunity, and so I'm about 3 times the national average on mine. It's time for another detox round on it, but it's important to know where you stand, because this chemical is one that is probably the most toxic, chronically toxic chemical that we've ever indiscriminately applied to the environment.
So
every aspect of the environment is contaminated with glyphosate.
You repeat that for me again, because I want to make sure we hear that.
Well, essentially, every aspect of our environment is contaminated with glyphosate. It's been so extensively used for so many years, with the promise that it's safe, that there's no problem. Very rapidly, poof, and it's gone, and all that type of malarkey that we've known isn't true for many years, but it's still promulgated and presented to people. You know, so safe you can drink it. Use correlations. You know, why it's safer than table salt. Well, that's not true. Anything can totally get, if you want. Yeah, you can drown 2 in water, but that's not the issue at hand. It's a chronic toxin that accumulates over time, but its effects are so extensive, so widespread, that it doesn't miss the system.
It's freakish when you think about it. When we look at soil, where with the extensive use that's been used in farming, with the runoff from soil, is the assumption made that it's also in the water supply?
No question. Not only in just the runoff water, but it's also in a lot of our deeper aquifers. so that even our groundwater is contaminated. If it gets down 3 or 4 feet in the soil, we'd refer to that as the vadose zone. That's a zone from there down that doesn't have biological activity. So that the only thing that's going to stop it from going into the water then, and you look at getting it down below that level, many of our roots, alfalfa roots, will go 20 feet deep. So that glyphosate that moves out of the in the root exudates doesn't have much exposure as far as microbial degradation. So it's either going to have to be chelated to tie it up, or it's going to move on down. It's a highly water soluble compound. It's very actively immobilized by your cations in soil. That's going to be your calcium, magnesium, iron, and manganese, and all of your mineral elements. But then you see the report from the University of Wisconsin last summer on eutrophication of Lake Erie. And what they're finding is for every acre of tillable land in the Lake Erie watershed, you have 1/3 of a pound of phosphorus as glyphosate moving into the lake. Well, that tells you that it's not being degraded to start with, but it's also they've already saturated the cation capacity in those soils and the filter strips for that water that it moves across before it gets to the lake. That area has already been saturated with glyphosate so that it can't contain anymore, can't stop any more movement. Well, some of the few organisms that can utilize glyphosate as a nutrient source are your blue-green algae. And so what you do is kill off all of your other vegetative competition and your microbial competition that would normally control that algae. And so we have all of these severe algal blooms and the red tides and those phenomena now because of the disruption of the environment that's brought about by that glyphosate moving into the water. And then you get all of the approaches for aquatic weed control of dumping glyphosate directly in the water, as they did in Yakima Valley and Yakima, Benton, and Hamilton counties in Washington state. And so 2 years after they started dumping that in the water for invasive weed control, all of a sudden you have this massive epidemic of anencephaly in children, where they normally born stillborn, or if you get to hold your baby, it's 24 or 48 hours because the brain isn't developed fully from that glyphosate effect.
It's wicked. So one thing that you just mentioned on that, so when we look at the agricultural capital of this country, the Midwest, and a lot of the watersheds are fed by, well, the Mississippi River Delta, or the Mississippi River coming through that. All the farming, like you were mentioning in Lake Erie, is dumping a lot of that glyphosate into the Mississippi River. So if we look at the flow going into the Gulf of Mexico, the red tide issues that we're seeing all throughout the Gulf, throughout Florida and such, you're saying there's a direct correlation with the glyphosate exposure that's killing the microbiome of the Gulf and killing the microbiome of the ocean systems as well, that's causing those red tides or causing those algae blooms.
Certainly a major contributing factor to it.
That's freakish.
No question. I think the University of Wisconsin study on Lake Erie is probably as good an example to give as you need for the following that through.
It's a step I didn't want to take, or a correlation I didn't want to draw, but that's really, really scary. So to look at remediation, because you jumped into my world talking about a detox program. What do you do currently? And I'm going to lead you into clinoptilolite, because I know I've read and I referenced some of the articles that you've been in talking about it. And you talk about the calcium, magnesium, potassium, being able to chelate glyphosate in some aspects, but it's the negative component of it chelating those essential minerals in the body. Clinoptilolite is one that, as a mineral, has the ability to chelate and trap and bind glyphosate, correct?
Yeah, with the clinoptilolite, you have that tetrahedron structure that is important in maintaining that and then getting the size down. But at Dana Stanley, Dr. Stanley, and Queensland, Australia, has some excellent research using the clinoptilolite then in poultry feed to remove not only the glyphosate, but it also removes the pathogenic salmonella and the clostridia, so that you get that entrapment and movement then out of the animal. So that her research was very important in removing the embargo against exporting Australian poultry products to Asia. And their concern was salmonella. Well, salmonella is a problem for us also.
Yeah.
And a lot of that is because of the glyphosate. You get the increased virulence, you change the gut microbiome that would normally suppress the enterococcal organisms that would suppress salmonella and the Clostridial species. And so it's important to remove both the glyphosate as well as get that microbiome reestablished. And then, as Dr. Stanley has shown, you also remove the salmonella and the clostridia pathogens while you're removing the glyphosate with the clinoptilolite. So in that situation, it's mixed with the feed. You'll see an increase in animals as far as fertility, and that's a concern. You and I are endangered species. Maybe me from age, but we're an endangered species as a whole, with 59% drop in sperm viability in the last 30 years. You see 30% drop in just the last 5 years of fertility in the population, which, you know, we're an endangered species there. I had a bull breeder that had to pull 40% of his bulls out of service because he couldn't get conception with the semen.
No kidding.
When he dusted his pastures with some humic, fulvic acid, and clinoptilolite, and in about 6 months, he restored fertility. You'll see the same thing in humans of couples that haven't been able to have a family. That many of those will find with the detox from the glyphosate that fertility is restored. And,
that's actually something I've seen, over the last couple of years of working with friends and associates, on both sides. A lot of women that are having fertility problems. The IVF, approach that people are taking. So you can direct, and I've been able to have great experiences with people using this little silica. It's my shameless plug for a second. We'll get into that because I want your feedback. And I haven't had an opportunity to share a product with you yet, which I'm going to send you, but I've seen it numerous times with women that haven't been able to conceive or were having major problems with miscarriages, being able to get pregnant, that weren't able to get pregnant, but then carrying to term, because it's the two components. You can get pregnant, but if you're so toxic that you're killing your fetus is one thing, but then also the conception aspects. And the same thing goes for men with sperm motility, like you're saying.
That issue got me in trouble with, got a lot of negative response when my private letter, a very confidential letter to Secretary Vilsack, was leaked to the public. When I had a group of veterinarians that I had the privilege of working with on an assignment, and they had identified the miscarriage issue and identified a new protein that we hadn't seen before. It's not in any of the protein databases and from a scientific standpoint. And when I wrote a letter, very confidential, I didn't even send it through the mail because I didn't want it compromised, that we were seeing associated with the GMO crops and with glyphosate, heavy glyphosate usage. So I used one of our intelligence couriers to deliver that directly to the secretary, and that letter was leaked. And of course, that's probably why you heard my name, because it was used in vain. I wrote that letter to a politician, not to a scientist, and so some of the terminology was a little different than most of my colleagues would anticipate coming from me. And I got phone calls wanting to know if this was a hoax, if I really wrote that letter. And I couldn't share the names of other people, because as soon as their names became known that they were involved with anything that might be negative or have a negative connotation for our new technologies, they lost their job. And that was part of the purge system that the companies have used to suppress or to censor a lot of the science that's been involved here. But still have some research going on in that area with the veterinarians. And I was in that situation. I was the messenger,
Yep.
not the cause on it. But the cause was there. There were correlations were good there to say, hey, this is something that we need to look at to understand pseudopregnancies and infertility. You can identify that with in the saliva with the scanning electron microscope. You'll see those entities. It responds all the criteria for a prion, and that it doesn't contain DNA or RNA. The veterinarians initially referred to it as a microfungus because that's the way it looked under the scanning electron microscope, as it would multiply in the tissues. You would get that kind of microfungus type of an impression. You had to blow it up about 40,000 times to see that. Additional research, we have a tremendous amount of information on it, where it occurs and what the conditions are. And again, your glyphosate is a major factor where we see that new protein being produced, especially with the GMO crops that are herbicide tolerant.
I'm one who grew up in a veterinary practice, so I love being able to bounce back and forth between the animal and human aspects. So what you're going over, and we know that the correlation between the two, we're not too far off. The miscarriage rates and everything that we're seeing in animals, you see in humans, it's a direct correlation.
Yeah, and if you look at those, do an SEM on the amniotic fluid or the placenta or that type of tissue, you'll just find this protein in really intense quantities. 2 months after that letter was released or was made public, the Chinese took some imported soybean seed from the U.S. and grew those plants up, took some scissors and cut one of the trifoliate leaves that had emerged and put that edge of the leaf on an electron microscope grid, and literally looked like you'd taken a 5-pound bag of sugar and spread it over the surface of the table. The concentration of that protein is so high. You'll find it in some of the pathogens. It's able to multiply there, and see it in saliva and other tissues with the miscarriage. Again, we haven't been able to pinpoint it as far as sequence, as far as characterizing it specifically. There has been one paper out by one of the veterinarians on it, just identifying a consistent protein, but not really going much further at that time, but still hoping to get that identification characterization completed so that we can publish the tremendous amount of epidemiological data and other information that we've been able to develop, and that's primarily the veterinarians, but been privileged to have an opportunity to work with them in that capacity. So
the great work comes from the veterinary medicine industry first. You have to have the foundation, because the medical establishment is not one to go down the wellness approach. But I can say that for my dad.
Well, once you get past that initial bias that this can't happen, and get a great person like Art Dunham in Iowa, or Ted Dupemeyer, that are willing to take that other step beyond and kind of willing to sacrifice some of their prestige and their reputation and say, hey, this thing is real and it's happening, and this is what we have to do to get over the hump if we're going to stay in business. And so they've been kind of in the forefront, but a number of others that have literally lost their jobs. You look at Dr. Judy Carmen as a result of her publishing and overseeing that study in Iowa with the pigs with Howard Vlieger. That cost her job in Australia because of the pressure from the industry. We could go through a number of other great scientists that literally sacrifice their reputation and their jobs because they're willing to be honest in their research. Many of them couldn't get funding to start with, and certainly after they published their data, all of them have essentially had difficulty with funding and maintaining their positions if they haven't been fired outright.
Well, and you were one of those guys as well, because you're a professor emeritus, and from my analysis, being at a major agricultural school that I would assume gets a lot of funding from agricultural sources, that one of them might produce a lot of glyphosate, that based on your work and everything you've done, you were offered the opportunity to retire early as a professor. So you couldn't continue to do what you were doing. Is my assumption in that correct?
No, I had excellent support from the dean, from the higher echelon. I had a new department head that as soon as that letter was leaked, he had called a national press conference so that he could disclaim any of my research for 35 years of ever being done at Purdue. And I found 6 colleagues in the department. The 3 of them were new after I'd actually retired, but I had very excellent. From Purdue, I was at a point where I could have retired at any time. I had, with Darrell Schultz, we had a major NSF grant that we were working on for some new technology following pathogenesis and disease development and things, but had a department head that wasn't happy with me. He was concerned about the 13 supported by industry, but as far as that, I had really excellent support. I had to do some accounting once in a while for some of my research, but as we discussed that, and I would say, well, okay, what are you asking me to do? And it was always keep up the good work. And so it wasn't like situation Barney Gordon found himself in Kansas, where He had to write a letter of retraction for publishing his research. He said, well, I wanted my growers, soybean growers, to know that the Roundup Ready soybeans were much less efficient in manganese. They had manganese deficiency anyway. And for them to have advance notice that to maintain production and production efficiency, they were going to have to look at some foliar feeding, which they weren't used to. Well, 2 months after he published that paper, he then had to write a letter of retraction. He said, I didn't understand the unintended consequences of publishing your research. And the pressure became great enough that he finally took a job outside the university. And that was too common a phenomena with Judy Carmen, Dr. Carmen, a tremendous international toxicologist. She was essentially fired from two or three different jobs on it, and blackballed from academia in those areas, as a number of other people have been that dared to challenge the system and report their study. You look at what Cirilini went through with his rat study at the University of Caen, 16 months after it was published, even probably the most peer-reviewed scientific paper that there's ever been, then having it redacted by the editorial board, and he went through the process of publishing again, and it was republished. But in the process, you get all of that ammunition for the industry. Well, it's been redacted, so it doesn't exist anymore. Well, that's not true. It's very, very valid study, very important study, but it also tells you why there aren't more studies that are out there, because of the serious consequences. But when you have an $8 billion industry, there's a lot of protection of that industry, even if it means people continue to die as a result of it.
Yeah, and we've seen that through hexavalent chromium and other areas as well. But this is orders of magnitude bigger than anything we've ever seen. Then, I mean, I didn't really understand the magnitude of this, from a thought process, because I've focused on heavy metals so much. But you can look at mercury being the worst neurotoxin known, and our exposure to mercury is pervasive and really, really bad. If you were to put a correlation between the two, if we looked at mercury, say mercury and aluminum, because of how pervasive they are, and we won't get into all the sources of those, for vaccinosis and such. But if you were to compare glyphosate with the two bad neurotoxins, glyphosate orders higher, because of our continued exposure. And I know they make each other worse. Where do you think they are?
Well, you have a lot of synergy between them. They're both out there, but you can avoid mercury pretty well, unless you're gonna be in an area where it's mined. And then, we prospect for mercury by flying over with a detector. So you have some volatility there, just like you have with glyphosate. But glyphosate, you don't have a choice on, because it's everywhere, and at levels that are highly consequential. But it's a chronic toxin, not an acute toxin.
And it
doesn't take a lot of mercury to be pretty acute, or even aluminum, or chromium 6. You get into those areas. So it's apples and oranges. You can say, well, which poison do I want? That's not the choice that you have in there. With glyphosate in our food supply, you have reduced nutrient density, you have antibiotic activity, you have the chronic aspects of chelation and all of those things that go on. So you don't want any of them, and there's no need for any of them. So I guess I don't don't get into that debate of what's most toxic, because
They're all bad.
why pick your poison?
Yeah.
Let's maintain the basic importance of agriculture as the basic infrastructure of society, and that's to provide that nutritious, high density, affordable, and economic and available food that we and our animals have to have for survival and for proper functioning. Let's do it. We can. We've done it for 2,000, 4,000 years. There's no excuse for what we're doing now with what we know. Doesn't mean we shouldn't use new science, new techniques, and that. But as someone said one time, as I remember growing up as a graduate student, that when we get into trouble in science is when we think that we've reached the plateau in utopia. And that's what happened with glyphosate. It was the silver bullet that was going to answer all the questions. And so with every incremental increase in progress, there are also some side effects, some unintended consequences. In science, if we address those, then we continue to make progress. If we do like we've done with glyphosate and stifle and censor and beat anyone that raises a question to death, then we go the opposite direction. It becomes a real anchor around our neck rather than a benefit to us. And we've seen that with the indiscriminate use of this chronic toxin that should have been very tightly regulated. And I've been involved in registration of several other chemicals. This one got a free pass for everything that we would normally require. It was just waived because it was supposedly so safe. They even wanted it approved for organic production because mammals don't have the EPSBS system. Well, we have the glutamate synthase and a whole lot of others that are also impacted very severely by glyphosate. So, and then you look at all of our neurochemistry originating through those 3 aromatic amino acids of phenylalanine, tryptophan, tyrosine that are produced in the gut by those microorganisms, because we don't have the shikimate pathway, but they do.
Yeah.
They're very sensitive to the antibiotic activity of glyphosate.
And that's where you've made some comments about dopamine production and serotonin production. You know, the glyphosate exposure really shuts off the body's mechanisms to do those effectively because of what you're talking about, the tyrosine and glutamate and such.
Yeah, that whole domino effect through all of those pathways.
So you could also draw a correlation with the lack of dopamine production because of the glyphosate issue, that a lot of our addiction issues and our drive to try to find that dopamine hit is also being spurred on because of our glyphosate exposure.
Right. It all ties together, and you can connect the biochemical dots directly in those processes.
So I want to get back to one point, and we'll wrap this up, and hopefully we'll be able to do this again, because I could do this every week with you and do an hour and go down the rabbit hole for people.
We're gonna have to do a lot of editing here.
This is completely unedited. I'm like good apple juice, you know, it's best unfiltered. And the point on clinoptilolite, a lot of the research has been done on the powders and a micronized powder for ruminants and such. And being able, like I sent you some of the analytical chemistry, being able to take clinoptilolite, maintain that honeycomb structure, but have fragments that are small enough to circulate through the body. You know, some of them may permeate cellular membranes and such. The benefit of clinoptilolite working systemically. Can you think of anything else that would be more beneficial for removal of glyphosate, or we can throw in, you know, the mercury, aluminum, and such as well, because they're all bound by clinoptilolite. Is there anything else you can think of? I know humics and fulvics are really good as well, but anything else besides that? I mean, is that one thing that
the
You know,
humic
could be a solution.
combination really work in synergy. And you have the, you have to get the clinoptilolite down to a size range that it can really move in the body or in the system to be really effective for it. So that that's one of the aspects, because it's not soluble.
Yeah.
You know, it depends on whether you have the calcium or the calcium potassium or potassium aluminum silicate that makes that crystal structure on it. So one thing there, where with the fulvic acid you have a soluble product that is going to move systemically anyway because of its solubility. But combination is really what's been found most effective in the studies that I've been involved in and aware of. Monica Kruger and all of her studies at Leipzig University was using primarily the humic fulvic materials, and very excellent success there. In fact, the European product that's available, the active almond, is the humic fulvic acid combination to remove it. Increases the removal with a little faster with clinoptilolite also there, or with Dana Stanley, where it's just a clinoptilolite then that is used as a component in the feed ration for the animals. But and the important thing is that it's removed, is that it's not bioaccumulating and not a persistent chronic problem for us.
And it's something from a removal standpoint that is a daily lifelong thing, because our exposure is not going to get any better based on what's in our environment.
Well, depends on if you're willing to change your diet a little bit. If you're willing to change your diet, one dose might do it. One thing to be aware of is that if you're going to take any of these products, whether it's humic or fulvic or clinoptilolite, any of them, you should be taking a good mineral supplement along with it, but not at the same time. You need some time, 4 or 6 hours or 12 hours between the two, or else you're just going to flush everything out that you're trying to complement from that standpoint. So that you realize that when you're detoxing, you're also removing a lot of beneficial nutrients in that process, and you need to be making sure that those are maintained, because they're essential nutrients.
Yeah.
So,
and with a lot of the binding sites on cells for the calcium, magnesium, potassium, the other essential electrolytes, you've got glyphosate that's binding to some of those sites, as well as some of the heavy metals. So the removal of the glyphosate and the metals, you've got to replace the good guys to go back to those binding sites so they get blocked again.
Right.
Okay. Well, I want to thank you, because we're a little over what we expected, but when we get to have fun, we can keep going down the rabbit hole. So I really appreciate the time. Hopefully we can do this again, and I'll send you some goodies as a thank you, and we will chat again shortly.
Well, I wish you the best, Clayton, and that approach, and certainly a very serious problem, and one that's not going to get any better unless we look at the remediation aspects.